We'll move on to the next section. It is the muscular system, and I want to, actually I'm not going to show you this clip just yet. I want to show you this picture first. When thinking about the muscular system, generally in your body there are three types of muscles. There's skeletal muscle. It attaches muscles to bones, or it attaches to bones, or your skeletal system I should say. It's also called striated muscle because it has a striped appearance on it, hence the name striated. There's also smooth muscle. This is the muscle that would line your digestive tract and allow for food to move via peristalsis along your digestive tract. Skeletal muscle, for the most part, you have to, it's under conscious control. Smooth muscle is not. You don't have to think to make your digestive tract work. And then a third type of muscle called cardiac muscle. And cardiac muscle is a little bit unique in that it has some appearances similar to both striated and smooth. You don't have to think to make your heart work, although you can, via thought processes, affect your heart rate. But we're going to key on skeletal muscle or striated muscle. We've already talked about heart muscle and smooth muscle in relation to some organ systems. So we're going to key on heart muscle, or on skeletal muscle. One of the things about skeletal muscle is that muscles can only get shorter. They can't get longer. And so you might say, well, how in the world can then you do this? Push a chair. How can you push something? How can you push something? Well, that's because this tricep right here, when I push something, the tricep shortens, okay? So the tricep muscle is look at that, man. My kids call those bingo wings, you know, these floppy things here. When the old ladies at the bingo parlor go, bingo! Those things start flapping, you know. Anyway, bingo wings. Yeah, so the tricep shortens, and that causes the muscle, the arm to do that. And so even though I'm pushing something away, the muscle involved in that is in fact shortening. It's pulling together. It's pulling this part of my forearm this way. So muscles can only shorten, and they're under voluntary control. So thinking of that voluntary control, here's an example of a malfunctioning of that system in goats. And you saw that up before. It's about fainting goats, and well, I'll just play the video and let you enjoy it.
Goats, the earliest known domesticated farm animal. Today there are more than 400 million of them worldwide. Then there's this little guy. He doesn't get the attention of, say, the more fashionable Angora goat. He's a fainting goat, and he's got a different talent. Lisa Johnson breeds goats in Florida.
These goats are called myotonic goats. Many people call them fainting goats, wooden leg goats, stiff leg goats, nervous goats, the Tennessee goats, Tennessee meat goats. So they just have all kinds of nicknames for them. They carry the hereditary gene for myotonia congenita. What happens is that when the goat is startled or excited, it causes a very temporary stiffening of the muscles. And when the muscles relax, after a few seconds, the goat jumps up and runs on its way.
They don't call them fainting goats for nothing.
It can even be something as simple as being excited over being fed. The adult animals typically they keep their balance better, and what they will end up doing is pretty much bracing on four legs.
While it might seem easy to make fun of the fainting goat, there are scores of breeders who love them just the way they are.
Okay, so hope you enjoyed that. Fainting goats. Glad you're not that way. Well, I want to do a little bit of thinking now about the structure of muscles and the nature of how they're put together, what their basic functional or structural units are. So down here in this picture, we have a bone. This is the radius, so one of the bones of the forearm. And there's a tendon here coming off the bone. Now we're going to learn, when we get to the skeletal system, there's a fibrous sheath, if you will, called the periosteum around bones that the muscle fiber is connected to. So I want you to catch here that the muscle is connected not just to one spot on the bone, but to this whole covering on the bone, that the connection is much more intimate than just being glued in one spot. So it's this whole covering. at any rate, this is the tendon, and this will sort of meld into the muscle fiber. And I do use that word muscle fiber because we don't really say muscle cells, because as you were developing in some of those early stages of development, the muscle cells that were in you sort of merged to become muscle fibers, and that's what we see right here. So here we have a whole muscle group, and there are different bunches of muscle fibers covered by a fascicle. If you've ever eaten a meat and gotten some gristle in it, if you will, some of that, you've seen some of this tough part of meat, that some of that can be fascicle, that's holding those bundles of muscle fibers together. And so then let's look at just one muscle fiber. And this is, you can kind of see these are the nuclei of all the muscle cells that fuse together to make a muscle fiber in development. So now we've got a muscle fiber, and inside of that muscle fiber, you can see all these little round structures. Those are called myofibrils, and it's inside of that we see the basic contractile unit of the muscle fiber. So we look at it a little more closely. So here now we have one muscle fiber. You can see that image I was looking at previously is shrunk down. It's right here. So we're looking now at one muscle fiber. So here's a muscle fiber, and inside of it are all those little round myofibrils. And if we look at one of those myofibrils, we're getting more and more microscopic here. We'll see inside of it a couple of key proteins, and they are actin and myosin. And here we have an actin protein. It's the thinner of the two, and myosin is the thicker of the two. Those collectively make up the bulk of your muscle tissue, actin and myosin. You can also sort of see an arrangement here. When we look at this myofibril, there's an arrangement of bands or stripes to it. Light areas, dark areas, that sort of thing. And here's an actual electron micrograph of a muscle fiber, and in reality, you can see those bands. So this is an artist's rendition. This is actually what it looks like. So we want to pay attention to that. And if we go from this one dark line right here to this dark line right here, they're called Z lines, that would be called one sarcomere. And this, you can make the argument, is the basic structural unit of the muscle. And it's the sarcomere, because this is what actually shortens. This is what shrinks together. And there's aspects of this that you see down here that actually move together when a muscle fiber is stimulated to do so. And so that's the basic structural unit, the sarcomere right there. These myofibrils, this muscle fiber, is covered by a membrane called the sarcolemma. So here's a little of that biojargon again. When you see a prefix, there's actually two of them for muscles. Myo would be one, or sarco would be another. Either of those two prefixes means you're probably talking about something that has to do with the muscle system, the muscular system. So this whole myofibril is covered with a membrane, the sarcolemma. And it's not just a covering, but there are parts of this that actually extend down into the group of myofibrils here. And you can see these little green and blue tubes that are extending down into there. Those are T-tubules that are actually connected to calcium storage sacs down inside of this. So the sarcolemma here is not just covering it, but it's intimately connected down deep into this muscle fiber to all the myofibrils so that it can communicate with them, tell them when to contract and when not to contract. Okay, there's a network here that we call the sarcoplasmic reticulum, and it's kind of like the system of tubes and cells we call endoplasmic reticulum, if you remember that. Endoplasmic reticulum, sometimes they were rough ER and they had ribosomes on them, and they were responsible for packaging and processing and moving proteins around. Smooth ER didn't have those, and they dealt with fats and things of that sort. So here we have sarcoplasmic reticulum, and they're connected to the sarcolemma. And so you get the idea that, again, we have a system of tubes here that's meant to carry something around, and it has to do with that calcium. It's got to move the calcium around. Calcium, now let's think about this. Calcium, we know it's important for bones, haven't gotten there yet. We know it's important for blood clotting. We know it's important for heartbeat. Now we're going to find out calcium is important for muscle contraction. So really essential mineral in your bodies. And its availability is an important consideration. Okay, so we've got the sarcoplasmic reticulum. We've got this covering. We've got all the structure, if you will, of the muscular system sort of right there on the screen. You can see it. Well, how does it work? Well, it's dependent on calcium, as I said, and it's also dependent on ATP. You probably would have assumed that. You know you spend energy when muscles move. Well, how does that happen? Well, there's a multi-step process, and your textbook talks about it. I'm going to show you a video clip in just a minute that illustrates it. I'll put it on Moodle as well. But I'm just going to go over it sort of briefly here, and I think I've got another image of it right here from your textbook. And really, folks, this is cyclical, so it kind of depends on where you want to start the process. This is showing 4 steps. I'm actually going to make it 6 steps. If you look at your notes, there's 6 steps there. The first thing that has to happen isn't even spoken of here on this screen. I got to go to another screen. Right here, this is represents a nerve, a neuron. And right here, there's an element of that neuron that's connecting to different muscle fibers. So each of these big round things would be a muscle fiber, and there'd be lots of little myofibrils inside of it. So if we look at this a little more closely down here, we can see that the end of that neuron is releasing a chemical that sort of falls on the muscle or is sensed by the muscle fiber. Actually, interestingly enough, the ability to get these little packets of chemicals to the muscle is also dependent on calcium. So that's a separate item. So these little messenger molecules, we're going to call them neurotransmitters. We go to this next picture over here. There's a cleft, a space between the neuron and the muscle fiber, and these little particles carry the message across that cleft or that opening to the muscle fiber, telling it to contract. Okay, so that's got to be the first step. So step number 1 in muscle contraction is for the message to go from a neuron to the muscle fiber, telling it to contract. And when that happens, that's a chemical transmission of that message, and it's going to go across the membrane, across the cleft there. Okay. When that happens, then we get to step number 2, and I got to go back to the previous image. Step number 2 is right here, and we're looking at the myosin molecule right here. And there's sort of a resting state where you have calcium bound to it. And when the calcium is bound to it, not the resting state, so resting state is right here. When calcium comes along, step number 2, it binds to it and it changes the shape of the myosin molecule. In the animation I'm going to show you in just a minute, you'll see that. Okay? So right here, the calcium binds to the myosin molecule and it changes, it twists a little, if you will, and it opens up a bonding site on the myosin molecule. So in your notes, you'll see step number 2, calcium is released, and that creates an open binding site on the actin molecule. Actually, it should be on the myosin molecule. It's misworded there. Oh no, yeah, okay. So then we get to step number 3, ATP, ADP. Transformation happens. Basically, energy is burned in the creation of ADP and phosphate, and what that really serves to do is cock the myosin head. And up here, if you look at this, we're looking over here. ATP goes to ADP, and basically it's pulling that myosin head back to the cocked position, and that's the only time that that energy actually gets used in the muscle contraction process. So there we actually have the conversion of ATP to ADP to pull back that little knob, if you will, there on the myosin molecule. And no, I got that. Yeah, on the myosin molecule, and make it ready to act. Okay. So then we get to step number 4 here, and in this step, because on the actin molecule, calcium came along and opened up a binding site there. Now, and that's what we're looking at here. Then this is the actin molecule. The myosin bonds to it because that site is opened up because of the presence of calcium. Myosin will bond to it. When myosin bonds to it, then pretty quickly it flexes. You see that down here. So here's the bonding. Here's the flexing happening, and the muscle actually will shorten when that happens. So that's going to happen, and then along comes a new ATP molecule. It's going to bond to the myosin head, and when it bonds to it, it's going to free it from the actin so that it can repeat the whole cycle all over again. So that's still images of it, and I've got a little short video clip I'm going to show you of basically an animation of this whole process happening.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so a little more detail there than I gave you in the 6-step process or is in your textbook. But if you stick with what you see here from your textbook, the process that I've given you, you'll be fine. One of the things I wanted to mention also, let me go back to another screen here. In this process, when stimulated to contract, a myofibril will contract completely or not at all. The muscle fiber actually will contract completely or not at all. So in this diagram up here, we've got a muscle group, and here's a myofibril. This would be a muscle fiber, and so that muscle fiber is going to, when it's received enough stimulus, a little bit of stimulus, it won't pay attention to. But when it crosses the threshold, it's enough to make it contract. It'll contract completely or not at all. So then I was thinking, okay, well then how do I, if I got to itch my nose like this, and if the muscles in my forearm and upper arm contract to allow me to do this, how do I keep from hitting myself in the face, if they contract completely or not at all? Well, the answer is muscle fibers contract completely or not at all, but a muscle is not made of just one muscle fiber. This whole muscle group is made of multiple muscle fibers. And so for any given movement, only a few of them or part of them will contract. Only on the most extreme instances will all of them contract. And so that allows us to have the fine motor movement that we have as people. And allows us not to punch ourselves in the face if we want to itch our nose. There's 3 or 4 ailments I wanted to just mention lastly here as I'm finishing up. This process whereby acetylcholine, ACH, that's what ACH stands for, acetylcholine. Oops, that's the release of acetylcholine here into the muscle fiber. Again, acetylcholine, ACH, is just it's a part of a family of chemicals called neurotransmitters, and its job is to make your muscles move. If you've ever heard of poison arrow frogs, the curare, that is used by some of the tribes in Amazonia in South America, it actually works by blocking this action right here and creates a paralysis because the acetylcholine doesn't ever stimulate a muscle. Another ailment I wanted to mention is muscular dystrophy. I might actually stop with that one. I might show you another video clip when we come back to lecture next week. So I'm going to hold off on muscular dystrophy for just a second. I'll mention 2 last ones though. In your notes I have rigor mortis and botulism both listed there. And if you think about rigor mortis, probably you would associate that with death, and you would be right. Rigor mortis is often used to describe that rigid condition that a body assumes after death. And it occurs because of the body's failure to produce any more ATP. Without ATP, you're not going to get the release here of the myosin from the actin, and so it stays bound and the muscle stays rigid. Well, this doesn't stay bound forever though, and so the ATP will eventually, or the bind between the actin and the myosin will eventually deteriorate, and the muscles will let go. And this is how coroners will determine time of death, by the rigidity of the body. 15 to 24 hours, somewhere in that ballpark, and bodies are no longer rigid because that bond is broken. So rigor mortis is used to evaluate time of death. And the last one is botulism. This is a botulinum toxin, and it works by preventing acetylcholine release. And so right here, curare blocks this process. Botulism prevents the release of acetylcholine right there. And I think I'm going to hold off on that one. I've got another little video, or not video, but at least a still image I want to show you of that. And so I think I'm going to stop right there, and hope this all works well. You're able to see these videos, and we'll pick it up with muscular dystrophy and botulism next Wednesday. So hopefully you do well on the exam tomorrow, and have a good day.